The year was 1856. A strange and costly crime was spreading across France. It was a silent thief, one that crept into the cellars of hard-working men. It targeted their most prized possessions: wine, beer. A winemaker would bottle a perfect vintage, full of promise and rich aroma. Weeks later, he would open it, only to find a sour, cloudy liquid that smelled foul. The drink was ruined, the profit lost. This was not an isolated incident. It was happening everywhere, from the grand vineyards of Bordeaux to the humble breweries of Lille. The nation's finest beverages were turning against their makers. No one knew why. This mystery baffled everyone. They needed an answer. They needed someone who could look at the problem differently, someone who could see what others missed. The call went out for an investigator of a scientific nature, a man who could solve the puzzle costing the nation a fortune. The country held its breath, waiting for a hero to unmask the invisible enemy. The man chosen was Louis Pasteur. He was not a detective in the traditional sense. He was a chemist, a professor. Known for meticulous work and unwavering curiosity, he approached the problem with a calm and methodical mind. He did not arrive with preconceived notions. Instead, he brought a simple yet powerful tool and an open mind. His job was to enter the world of these spoiled drinks, examine the evidence up close, and find the truth, no matter how small or unbelievable it might seem. The case of the sour wine had found its lead investigator. Louis Pasteur arrived at the crime scenes with none of the usual tools of an investigator. He carried no magnifying glass for fingerprints, no notebook for witness statements. His primary weapon was a microscope. At the time, this instrument was not widely used for such practical problems. It was mostly a tool for academics observing pond water, observing dust. But Pasteur believed the answer to this great mystery was too small to be seen with the naked eye. He suspected the culprit was hiding in plain sight, invisibly within the very liquid it was destroying. He was about to introduce the world to a new kind of detective work. He began his work methodically. He took a single drop of good, healthy wine. He placed it carefully on a small, clean slide of glass. Peering through the eyepiece, he saw what he expected: tiny, round yeast cells. These were the known agents of fermentation, helpful organisms that turn sugar into alcohol. They floated peacefully in the clear liquid. This was the baseline, the picture of a beverage in good health. It was the control group, the scene before the crime had taken place. He documented his findings with precise drawings. Next, Pasteur turned to the spoiled wine. He took a drop from a cloudy, sour bottle. He placed it under the same lens, adjusting the focus with a steady hand. What he saw was dramatically different. Alongside the familiar yeast cells, there was something else. Tiny, rod-shaped organisms swarmed through the liquid. They were smaller than the yeast and moved with purpose. He saw them in every sample of bad wine, in every bottle of bitter beer. The good drinks had only yeast. The bad drinks had these other unwelcome invaders. The pattern was undeniable. To Pasteur, the conclusion was as clear as the unspoiled wine. These tiny rods were the intruders. They were the vandals responsible for the sour taste and the foul smell. He called them microbes, a general term for microscopic living things. Presenting his findings was the first major obstacle. Pasteur stood before his scientific peers, declared that living creatures, invisible to the naked eye, were ruining the nation's wine. The idea was met with deep skepticism. At the time, the prevailing theory was spontaneous generation. Most scientists believed that life simply appeared from non-living matter, like maggots on rotting meat, mold on old bread. They argued that the microbes Pasteur saw were the result of the spoilage, not the cause. They thought the wine went bad on its own, and these tiny organisms were just a byproduct of the decay. This wall of disbelief was formidable. His colleagues, respected men of science, dismissed his theory as fanciful. They challenged him publicly, demanding more proof. They argued that air itself, Some unknown chemical imbalance was the true culprit. Proving that something so small could have such a massive economic impact was a revolutionary idea. Revolutions are rarely welcomed with open arms. Pasteur was not just fighting a scientific mystery, he was fighting the established dogma of his time. He knew that observation alone was not enough. He needed to conduct an experiment that would prove his theory beyond all doubt. His next challenge was a practical one: how to eliminate the unwanted microbes without destroying the drink itself. His first idea was simple: heat. He knew that boiling water killed living things, so he tried boiling a flask of wine. The experiment worked in a way. After boiling, he sealed the flask and the wine no longer soured. The rod-shaped microbes were gone. This was a crucial piece of evidence. It demonstrated that killing the microbes prevented the spoilage, but it also created a new problem. The intense heat of a full boil completely ruined the delicate flavor and aroma of the wine. It tasted cooked and flat. This led to the third major hurdle: inconsistency. The method that worked for one beverage failed for another. Wine was delicate. Beer was heartier, but could still develop off flavors if overheated. Milk, another beverage prone to quick spoilage, presented its own unique challenges. A single solution would not work for all. Furthermore, the heating process itself was difficult to manage. Some microbes were more resilient than others, surviving a brief warming only to multiply again later. The results were inconsistent and unreliable. Pasteur realized he needed more than just heat. He needed precision. The question was no longer if heat could work, but how much heat was just right. With the knowledge that heat was the weapon but a full boil was too destructive, Pasteur began his most important series of experiments. He approached the problem like a master locksmith trying to find the right combination to a complex lock. He needed to find the exact temperature that would kill the harmful microbes while leaving the beneficial qualities of the drink unharmed. This required a level of precision unheard of at the time. The tools for measuring temperature were crude and often inaccurate. A few degrees difference could ruin an entire batch. So Pasteur became an inventor as well as a scientist. He had to design and build new equipment, flasks, thermometers, capable of maintaining and recording heat with greater accuracy. His laboratory became a workshop of glass, metal, flame. He would take dozens of samples of the same wine. He would heat each one to a slightly different temperature: 50 degrees Celsius, 55 degrees, 60 degrees. He would hold them at that temperature for a specific length of time. It was slow, painstaking work. Each test required patience and meticulous record-keeping. He was hunting for a very narrow window of opportunity. After heating each sample, he would cool it down and set it aside. Then, the waiting began. He observed the samples for days, weeks. He used his microscope to check for any surviving rod-shaped microbes. He also performed the most important test of all, the taste test. He tasted the wine himself. Panels of professional tasters sampled the treated wine. Was the flavor intact? Was the bouquet still rich and appealing? He noted how a wine heated to 65 degrees lost some subtle notes. He noted how one heated to 50 degrees might still show signs of spoilage after a month. Through endless repetition, a pattern finally emerged from his detailed notes. He discovered the magic number. For wine, heating it to approximately 55 to 60 degrees Celsius, about 130 to 140 degrees Fahrenheit, for a few minutes was enough. This gentle heat was sufficient to kill the spoilage-causing microbes without cooking the wine or boiling away its soul. The flavor remained almost perfectly preserved. The drink was stabilized. It would no longer turn sour. He had found the delicate balance between safety and quality. He had solved the central puzzle of the investigation. Pasteur had found the solution. This gentle heating process, just enough to eliminate harmful bacteria without ruining the product, was a triumph. The method was soon named pasteurization.